Have you ever stopped to wonder what's really going on inside your cells when your body faces some kind of challenge?
Yeah, like a sudden burst of exercise or maybe chronic stress from deadlines.
Exactly. Or even just, a persistent cough.
Yeah.
Your cell. They aren't just passive bystanders, not at all. They're actually masters of adaptation, constantly adjusting to survive and, well, thrive.
hmm
So today we're taking a really deep dive into cellular adaptations, these incredible, intricate ways cells change in response to stress or increased demand or even injury.
That's right. And our mission today really is to arm you, our budding medical professional, with a practical, memorable understanding of these absolutely fundamental processes.
They want to make it stick.
Exactly. We'll simplify the complex stuff, give you tangible examples, and maybe throw in a few anchors, some memory aids, maybe some questions to get you thinking, to help you really understand, retain, and recall the crucial details.
Think of it as your essential cellular survival guide.
That's a good way to put it.
Yeah. We're going to explore the how behind cell growth and change, starting with how they talk to each other, their communication networks, then the powerful concept of the cell cycle.
Right. And then we'll get into the 5 main ways cells adapt, and crucially, what happens when those adaptations maybe aren't enough, or even worse, when they start to go a bit sideways, sometimes paving the way for disease.
Okay, let's unpack this then. Some foundational concepts first. Before a cell can adapt, it needs to know what's happening around it, right?
Absolutely.
So what's the very first domino to fall? What kicks off that change, and how do cells actually, talk to each other?
It really all starts with chemical signals. Signals from their immediate neighborhood, their microenvironment. Cells and, well, any multicellular organism are constantly communicating, sending messages, receiving messages.
Okay.
These signals could be hormones or local mediators, sometimes even direct cell-to-cell contact. Now, to simplify how they talk these intercellular signaling mechanisms, let's use the APE analogy, APE, for the 3 main types.
APE. Okay, I like analogies. What's the A?
So first, autocrine signaling, that's the A. This is where a cell literally talks to itself.
Huh. Like sending yourself a text.
Kind of. Yeah, like sending a text to yourself to remember something important. The cell spits out a signal that then binds to receptors right there on its own surface. It influences its own behavior.
Got it. Autocrine self-talk. What's P?
Next is paracrine the P. Here, cells talk to their neighbors, nearby cells, usually within the same organ.
So like chatting over the cubicle wall.
Exactly like that. A quick chat with a colleague nearby, short distance, local impact. The signal only acts on cells right there in the immediate vicinity.
Okay, auto-para. Must be E next.
And finally, endocrine the E. Now, this is the long-distance call. Hormones made by endocrine organs travel all the way through the bloodstream to reach target cells that might be quite far from where the signal started.
Ah, like a company-wide memo from HQ.
Perfect analogy. Reaching offices all over, influencing specific teams or cells.
So APE, autocrine, paracrine, endocrine. Different ranges of communication.
Precisely.
Yeah.
Now, despite all the, biochemical complexity behind these signals, the ultimate outcomes for a cell that receives one, they're surprisingly limited, but absolutely critical for its survival and function. Basically, 4 things can happen.
Okay, what are they?
A cell can survive, basically, resist programmed cell death, apoptosis. It can divide, meaning it enters the cell cycle. It can differentiate, specialize into a specific cell type. Or if it's maybe damaged beyond repair or just not needed anymore, it can die, again, through apoptosis.
Survive, divide, differentiate, or die. The 4 fates dictated by signals.
You got it. It's a powerful form of biological control.
And part of that signaling involves these molecules you mentioned earlier, growth factors. They sound important. What's the real insight there? Is it just about growth?
Well, they are key players, absolutely. You can think of them as sort of local hormones. They're polypeptide messengers, and they're coded by genes called proto-oncogenes.
Proto-oncogenes sounds potentially problematic.
It can be. What's fascinating is how these proto-oncogenes, when they're working normally, they act like the cell's accelerator pedal, promoting growth and division when needed.
Okay.
But if a mutation happens, it can turn them into oncogenes, and that's like having the gas pedal stuck to the floor. It drives uncontrolled cell growth, the kind you often see in tumors.
Wow. So fine line between control and chaos.
A very fine line. And these growth factors, they don't just stimulate growth, they really orchestrate the whole repair process. Imagine trying to build a house without a foreman, you'd get chaos, right?
Right. Stuff built in the wrong order or just uncontrolled expansion.
Exactly. That's what happens when these signals are out of sync. Uncontrolled growth or maybe stalled healing. Let's look at a few key examples real quick.
Okay.
EGF, epidermal growth factor, crucial for skin repair, liver regeneration. It gets epithelial cells, hepatocytes, fibroblasts dividing.
EGF, skin and liver. Got it.
Then VEGF, vascular endothelial growth factor. I think V for vascular. This one's a potent inducer of blood vessel development, vasculogenesis, and also the growth of new blood vessels, angiogenesis. Vital in wound healing, but also, alarmingly, in helping tumors get their blood supply.
Ah, double-edged sword there too.
Definitely. Then PDGF, platelet-derived growth factor, think P for platelet, released when platelets activate, big role in wound healing, recruits fibroblasts, smooth muscle cells.
Okay, PDGF platelets, wound healing.
And finally, GCSF, granulocyte colony stimulating factor, G for granulocytes. This one stimulates bone marrow, especially if it's poorly functioning, say, after chemo, to boost granulocyte production.
EGF, VEGF, PDGF, GCSF, each with a specific job in growth and repair.
Precisely orchestrated.
So, okay, let's say a cell gets the divide message from these signals, these growth factors. It gets the green light. What happens next? It enters the cell cycle, you said. Tell us about this cellular production line.
Gladly. The cell cycle is exactly that. A highly regulated sequence of events leading to cell division, making two daughter cells from one. It has 4 main phases, usually remembered by their initials.
Okay, lay them out for us.
First is G1, gap 1. This is the initial growth phase. The cell synthesizes proteins, gets bigger, kind of gathers its resources for the big job ahead.
G1, growth and resource gathering.
Then comes S, synthesis. This is the critical phase where DNA synthesis happens. The cell has to replicate its entire genome, all its DNA, perfectly. No mistakes allowed here.
S for synthesis. Makes sense. What's after S?
G2, cap2. More growth here, plus synthesis of proteins and organelles needed for division. It's also like a final quality check, double-checking the replicated DNA, getting everything ready for the split.
G2, final prep and checks, and then the main event.
And then M, mitosis. This is the mitosis or multiplication phase. Here, the cell divides its nucleus first, then its cytoplasm, splitting into 2 genetically identical daughter cells.
G1, S, G2, M. The cycle of division. What about cells that aren't dividing?
Good question. Cells that aren't actively dividing are said to be chilling out in G0. That's the resting phase or off cycle. They've basically stepped out of the cycle either temporarily or sometimes permanently.
So they can just sit there in G0.
Many do, but the cycle itself, G1 through M, isn't just a free-for-all. It's incredibly tightly controlled by key checkpoints. Think of them like quality control stops on the production line.
Quality control. Important for preventing errors, I imagine.
Absolutely critical. The most famous one is probably the restriction R point, late in G1. Once a cell passes this point of no return, it's generally committed. It will complete the entire cycle.
No turning back after the R point.
Pretty much. And these checkpoints, they ensure one phase doesn't start until the previous one is properly finished. And crucially, they detect damage, especially DNA damage.
Okay, so who's in charge of these checkpoints? Who are the regulators making sure everything runs smoothly?
The primary engineers driving this whole process are proteins called cyclins and their partner enzymes, the cyclin-dependent kinases, or CDKs.
Cyclins and CDKs? Heard of them?
Right. CDKs are inactive on their own. They need to bind to a specific cyclin to become active. These activated cyclin CDK complexes then phosphorylate other proteins, basically pushing the cell through the cycle phases.
So they're like the engine and the ignition key.
That's a great way to think about it. And there are key breaks, too. A critical one is the RB protein retinoblastoma susceptibility protein. RB normally acts as an inhibitor, a brake, stopping the cell cycle progression, especially at that R point.
So RB puts the brakes on. How does the cell get past it?
To move forward, RB needs to be phosphorylated, usually by those cyclin CDK complexes. Phosphorylation inactivates RB, releases the brake, and lets the cycle proceed.
Okay, so phosphorylate RB to go. What other guards are there?
Then there's the really famous one, P53, often called the guardian of the genome.
Guardian of the genome. Sounds important.
Hugely important. If DNA damage is detected at a checkpoint, P53 steps in. It can halt the cycle to allow time for DNA repair. Or, if the damage is just too severe, P53 can trigger apoptosis-programmed cell death to prevent that damaged cell from replicating and potentially becoming cancerous.
Wow, so P53 is a major failsafe. Halt, repair, or die.
Exactly. It's a vital defense mechanism.
So the cell cycle is this tightly controlled dance with accelerators and brakes. But you mentioned apoptosis. If the damage is too bad, P53 might make the cell self-destruct.
Right.
But what about those cells we rely on, like critical heart muscle cells. They're so vital they probably can't just afford to die or even divide easily when faced with stress. So how do they adapt to increased demand if they can't just make more of themselves? Keep that question in mind.
That's a fantastic question, and it leads us perfectly into another crucial concept. Not all cells have the same ability to divide. Back in 1894, a pathologist, Giulio Bizzarro, gave us a framework. We now apply it more broadly to tissues, keeping in mind the role of stem cells. Let's use the LSP framework to categorize them. LSP.
LST. Okay, what does that stand for?
First, labile L cell populations. Think of these as the high turnover crew.
High turnover crew. Like what?
They're continuously dividing throughout life, always regenerating. Their stem cells are constantly active, replacing cells that are lost. Your skin epidermis, for example, turns over about every month. The lining of your gut, even faster, maybe every 24 hours.
Wow.
And of course, bone marrow, cranking out blood cells constantly. That's labile tissue.
Okay, L is labile, always dividing. What's S?
Next, stable S cell populations. These are like the on-call specialists.
On-call specialist, meaning?
Normally, these cells are quiescent. They're sitting quietly in that G0 resting phase we talked about. But, and this is key, if they get the right stimulus, the right growth signals, they can re-enter the cell cycle and divide when needed.
So they're resting, but ready to go if called upon.
Exactly. Their stem cells are usually quiet too, or maybe slow dividing, but they can ramp up proliferation when required. The classic example, liver hepatocytes.
The liver. Famous for regeneration.
Absolutely. That amazing regenerative capacity, tied to the Greek myth of Prometheus, whose liver was eaten daily and regrew, that's stable tissue in action. Other examples are bone cells, osteoblasts, and kidney cells.
Okay, L for labile, S for stable. That leaves P.
And finally, permanent P cell populations. These are the terminally differentiated veterans.
Terminally differentiated veterans. They sound like they're done dividing.
They pretty much are. They've definitively left the cell cycle. If you lose a significant number of these cells, they generally cannot divide or replicate effectively to replace the loss.
So damage is, permanent?
Largely, yes. Stem cells might be hanging around in some of these tissues, but they just can't mount an effective proliferative response to large-scale damage. Think brain neurons, no regeneration in humans after injury. Cardiac muscle cells damage heals with a scar, not new muscle. Skeletal muscle has only very, very limited regeneration capacity.
Brain, heart, most muscle. Permanent. That makes sense why injuries there are so serious.
It absolutely does. Understanding these LS categories is crucial. It tells you which tissues can bounce back easily from injury and which ones are much more vulnerable to lasting damage.
It's like knowing which parts of your car are easily swapped out versus which ones mean a major, maybe impossible repair.
Precisely. Good analogy.
Okay, so now we know how cells talk, APE, how they decide to divide, cell cycle, checkpoints, and their inherent capacity for division, LSP. Let's get to the core. The 5 main ways cells actually adapt to stress. These are the big ones, right?
These are the big 5, yes. The ones you'll encounter constantly in medicine. We can remember them with the acronym RHHA, regeneration, hyperplasia, hypertrophy, atrophy, and metaplasia.
RHHA, okay.
And a key point before we dive in, these adaptations are generally reversible. They're changes in cell size, number, or even their type or phenotype, all happening in response to some change in their environment or demand. The cell is trying to cope.
Reversible coping mechanisms. Yeah, got it. Where do we start with RHA?
Let's start with 1, regeneration.
Okay.
This is basically the replacement of lost cells by identical cells. The goal is simply to maintain the original tissue or organ size after some loss.
So replacing like with like.
Exactly. Now, the capacity for regeneration varies. We mentioned human cells have a limited number of divisions, right? The Hayflick number around 61.3 times.
Linked to telomeres.
Yes. Linked to those telomeres, the protective caps on chromosomes that shorten with each division, like a cellular clock ticking down. Oh, and it's important to distinguish regeneration, replacing cells, from reconstitution. That's replacing a whole lost body part, like a lizard's tail. We don't really do that.
Right, no tail regrowth for us.
Unfortunately not. But great examples of regeneration in adults are the liver after you remove a piece, a partial lobectomy, or your skin epidermis healing after a burn.
Okay, regeneration, replacing lost cells identically.
Yeah.
What's the first H in RHHM?
The first H is 2, hyperplasia.
Hyperplasia, what's that?
This is an increase in the size of a tissue or organ, specifically because of an increased number of cells, more cells.
So this one will require cell division.
Absolutely. Which means hyperplasia can only happen in those labile or stable cell populations we just talked about, the ones capable of dividing. Permanent cells can't do hyperplasia.
Makes sense.
It's usually a controlled process, typically reversible if the stimulus is removed. And it can be physiological, normal, or pathological disease related. Sure, physiological hyperplasia. Think of the uterine endometrium thickening during the menstrual cycle or pregnancy. Pathological, maybe a thyroid goiter developing due to iodine deficiency, or, very commonly in older men, benign prostatic hyperplasia, BPH.
Okay, uterine lining, thyroid goiter, prostate enlargement. Got it.
Now, here's a really critical insight, a link to watch out for. Repeated cell divisions, which is what hyperplasia is all about, inherently expose cells to a higher risk of mutations during DNA replication.
More chances for mistakes.
Exactly. And this increased risk forms a crucial link between hyperplasia and neoplasia that's new, abnormal growth, like cancer. It's a path you'll explore more later, but the connection often starts here.
Right, so hyperplasia, more cells, more division, more mutation risk. A potential slippery slope.
You've got it.
Okay, that was the first stage. This brings us back to that question I posed earlier. What about those non-dividing cells, the permanent ones?
Yeah.
How do they cope with increased demand if they can't do hyperplasia?
Excellent recall. And that brings us perfectly to the second H in RHHAM, 3, hypertrophy.
Hypertrophy. Okay, how is this different?
Hypertrophy is also an increase in tissue or organ size, but this time it's due to an increase in the size of the individual cells, not an increase in their number. The cells themselves get bigger.
Uh-huh. So this is how permanent cells adapt.
Precisely. This is the main way cells like cardiac muscle or skeletal muscle adapt to increased functional demand. They can't make more of themselves easily, so they bulk up.
Like a bodybuilder's muscles.
Exactly. That's physiological hypertrophy of skeletal muscle. The pregnant uterus is actually a great example of both hypertrophy and hyperplasia happening together. The muscle cells get bigger and more are made.
Interesting mix. What about pathological hypertrophy?
Well, think about cardiac muscle again, maybe in someone with high blood pressure, hypertension. The heart muscle has to work harder, so the cells enlarge, the heart wall thickens. But here's where we need that anchor phrase. Bigger is not always better.
Why not? Bigger heart sounds stronger.
Not necessarily. In pathological cardiac hypertrophy, the muscle cells outgrow their blood supply. The capillaries don't increase proportionally, so you get relative lack of oxygen, anoxia, you can get fibrosis or scarring, and eventually the overworked, underfed muscle can fail. Myocardial exhaustion.
Wow. Bigger is not always better. That's a really important anchor for the heart. Okay, RHH. Next is A.
A stands for 4. Atrophy.
Atrophy. Sounds like the opposite of growth.
It is. Atrophy is the shrinkage of a tissue or organ due to an acquired decrease in the size and/or the number of its cells. The word itself comes from Greek atrophia, meaning lack of food, which points to one potential cause.
So cells shrink or die off.
Both can happen. At the cellular level, it often involves the cell shrinking, losing substance, getting down to a size where survival is still possible with fewer resources. At the organ level, it's usually a combination of the cellular shrinkage and apoptosis, programmed cell death, reducing cell numbers.
How do cells actually shrink themselves?
A key mechanism is increased protein degradation via the ubiquitin proteasome pathway. Think of this as the cell's sophisticated recycling plant. It tags unwanted or damaged proteins with ubiquitin, sending them to the proteasome for breakdown.
Okay, breaking down internal components.
Right. And as they break things down, sometimes undigested material accumulates in residual bodies. These often contain a pigment called lipofuscin, which you can sometimes see under the microscope. They look like little cell rubbish bins.
Cell rubbish bins. Lipofuscin. Got it.
Examples of atrophy.
Sure. Physiologically, think of ovarian atrophy after menopause, or the thymus gland undergoes involution. That's a specific term for physiological atrophy driven mainly by apoptosis shrinking dramatically during adolescence.
Yeah.
Pathologically, muscle atrophy from disuse is a big one, like if your arm is in a cast.
Muscles waste away if you don't use them.
Exactly. Or denervation atrophy if the nerve supply to a muscle is cut off. Right. Now, this reminds me of that earlier question I threw out there. Astronauts in space or patients bedridden for a long time, their bones get thinner. Why does that happen? And crucially, how is losing bone mass different from losing bone calcium?
Ah, yes, excellent connection. Bone atrophy, which we call osteoporosis, is exactly that loss of bone mass. It's often triggered by a lack of mechanical stress. Bones need that physical loading to maintain their density. Astronauts lose gravity, bedridden patients lose weight-bearing activity.
So lack of stress causes the bone tissue itself to shrink or reduce.
Correct. That's different from, say, a deficiency in vitamin D or calcium itself. That leads to conditions like rickets in kids or osteomalacia in adults where the bone matrix is there, but it's poorly mineralized. It's soft bone, not necessarily less bone mass initially. Different mechanisms, different pathologies.
That distinction is really important. Osteoporosis versus osteomalacia. Got it. Can atrophy be reversed?
Sometimes, yes. If you take the cast off your arm and start using the muscle, it can hypertrophy again. There's even medication like minoxidil that can reverse some hair follicle atrophy and baldness. But if atrophy goes on too long, especially if parenchymal cells, the main functional cells, are lost via apoptosis, reversal becomes much harder, often impossible.
Use it or lose it, basically.
That often applies, yes.
Okay, RHHA. That leaves M, the last pillar.
The last one is 5, metaplasia.
Metaplasia sounds complex.
It's a fascinating one. Metaplasia is a reversible change where one differentiated adult cell type is replaced by another differentiated adult cell type.
So one cell type literally changes into another.
Well, not quite the existing cells changing. It's more like an abnormal regeneration. It happens because the resident stem cells are reprogrammed. Under chronic stress, they differentiate along a different pathway, producing a new, usually tougher cell type instead of the original one.
Ah, the stem cells change their output. Why would they do that?
It's usually adaptive. The goal is to replace cells that are sensitive to a particular stress with a cell type that's better able to withstand that stress.
Can you give an example?
The classic one is in the airways of smokers. The normal lining is a delicate pseudostratified ciliated columnar epithelium, great for trapping gunk and sweeping it out with cilia. But cigarette smoke is harsh.
Right.
So under that chronic irritation, the stem cells switch production. They start making a tougher, more resistant stratified squamous epithelium, like you have on your skin.
Okay, tougher sounds good.
It's more resistant to the smoke, yes, but there's a trade-off. You lose the cilia, you lose the mucus-secreting cells. So you lose that cleaning mechanism, which increases the risk of infections.
Ah, adaptation with a cost. Another example.
Another well-known one is Barrett's esophagus. Chronic acid reflux from the stomach irritates the normal squamous epithelium of the lower esophagus, so it undergoes metaplasia, changing into a glandular intestinal type epithelium, which is more resistant to acid.
Squamous to glandular in the esophagus due to acid.
Got it. But again, there's a catch.
This sounds like another double-edged sword situation.
Precisely. That is the perfect anchor phrase for metaplasia, the double-edged sword. While it can be adaptive, providing protection, this metaplastic change, especially in epithelia, is often considered a potential stepping stone.
A stepping stone to what?
To dysplasia, that's abnormal maturation of cells, often precancerous and potentially even to outright cancer, carcinoma.
So why? Why does this adaptive change increase cancer risk?
The thinking is that the reprogramming of those stem cells, while adaptive initially, can lead to genetic instability. And the increased cell proliferation often needed to establish the new metaplastic lining provides more opportunities for mutations to occur and accumulate. It basically creates fertile ground for malignant transformation.
So the adaptation itself, while solving one problem, might create a pathway to a much bigger one down the line.
That's the significant clinical concern with many types of metaplasia, yes.
Wow. RHM. Regeneration, hyperplasia, hypertrophy, atrophy, metaplasia. What an incredible journey through how cells cope. It's clear they're always trying to find balance and survive.
They really are, constantly adjusting.
But before we wrap up, I know pathology has a few other tricky terms that often get confused with these, or with each other. Can we quickly clarify some of those important distinctions?
Absolutely. Good idea. While RHM covers the main adaptations of existing tissues, there are related terms about development and abnormality that are crucial.
Okay, what should we know?
First, aplasia. This means the complete failure of an organ or tissue to develop at all during embryonic development. It was never there to begin with.
So, born without it.
Essentially, yes, it's a congenital disorder. Think of thymic aplasia leading to severe immune deficiency, or maybe aplasia of one kidney. Often, if one kidney is missing, the other undergoes compensatory hypertrophy to pick up the slack.
Aplasia, complete failure to develop.
Yeah.
What about partial failure?
That would be hypoplasia. This is underdevelopment or incomplete development of an organ or tissue, again, during the embryonic stage. So the organ is there, but it's too small because it didn't form with enough cells.
Okay, so aplasia is none, hypoplasia is too little from birth. How is hyperplasia different from atrophy then? They both mean small.
Crucial distinction. Hypoplasia is a developmental issue. It never reached full size. Atrophy is an acquired condition. The organ or tissue developed normally, but then shrank later due to some cause.
Got it. Hypoplasia never grew enough. Atrophy grew fine, then shrank. Example of hypoplasia.
Testicular hypoplasia in conditions like Klinefelter syndrome.
Okay, aplasia, hypoplasia. What else trips people up?
The really tricky one, dysplasia. This word is a minefield because it has two completely different meanings in pathology. You absolutely have to rely on the context.
Two meanings for one word. Okay, that's confusing. What are they?
Meaning number one, abnormal maturation of cells within a tissue. This is the one we touched on with metaplasia. It involves cells that look abnormal variations in size, shape, nuclear changes, disorderly arrangement. This type of dysplasia is often precancerous, though potentially reversible.
So, abnormal-looking cells, possibly precancerous. Examples.
Cervical dysplasia, detected on a pap smear. Or carcinoma in situ, which is basically a very severe form of dysplasia where the abnormal cells fill the entire thickness of the epithelium but haven't broken through the basement membrane yet. Still technically pre-invasive cancer.
Okay, dysplasia, meaning one. Abnormal cell maturation, precancerous potential. What's meaning two?
Meaning number two, abnormal development or malformation of an organ. This is a congenital or developmental abnormality, similar to aplasia or hypoplasia, but referring more to disorganized development.
So, meaning two is about abnormal organ formation from the start.
Exactly. Examples would be renal dysplasia, where the kidney structure is malformed, or fibrous dysplasia of bone, a developmental bone disorder. Nothing directly to do with cancer risk in this meaning.
Wow. Okay, context is absolutely key for dysplasia. Abnormal cells versus abnormal organ development.
You absolutely must check the context. And finally, just a quick recap on necrosis versus apoptosis, the two main ways cells die.
Right, cell death. Important distinction, too.
Apoptosis, remember, that's the programmed, usually physiological, tidy cell death. The cell shrinks, collapses neatly, gets cleaned up, usually no inflammation. It's an organized self-destruct.
Apoptosis, programmed, clean death.
Necrosis, on the other hand, that's messy, gross tissue death. It's usually caused by external injury, lack of blood supply, ischemia, toxins, trauma. The cells swell and burst, spilling their contents, and it does trigger an inflammatory reaction, much less controlled.
Necrosis, messy. Injury-induced death with inflammation.
Perfect. Those distinctions are fundamental.
They really are. That clarifies so much. This deep dive has truly laid out an essential toolkit for understanding how our cells navigate stress, injury, and just life.
Indeed. And just to reinforce the main message, cellular adaptations, RHAM, are generally reversible changes, allowing cells to survive and function under stress. We saw hypertrophy for workload in non-dividing cells.
Right, getting bigger.
Hyperplasia for increasing cell numbers in dividing population.
More cells, but watch that mutation risk.
Atrophy for shrinkage when demand decreases or nutrients are low.
Use it or lose it.
And metaplasia, that switch in cell identity, the double-edged sword, often adaptive, but carrying that risk of progression. And understanding that LSP classification, labile, stable, permanent, helps predict which adaptation is even possible for a given tissue. Plus, now you've got those key distinctions for aplasia, hypoplasia, and the two faces of dysplasia sorted.
Absolutely. And these aren't just, academic concepts you memorize for an exam. These are the fundamental processes that underpin health and disease. These are the very things you'll be observing, diagnosing, and hopefully treating in your future patients.
Precisely said. They are truly the language of disease and recovery at the cellular level.
So as you, our listener, go about your day after this, maybe take a second to think about your own body, your muscles, your skin, your internal organs. What silent cellular adaptations might be happening right now, keeping you in balance? Or perhaps subtly shifting things towards a new state?
Yeah, and how does our modern lifestyle, our stress levels, our environment, how does it continually test these ancient, fundamental cellular survival strategies? Something to ponder.